There's a really good reason for things, oftentimes when you look back in history. There's a reason that we thought the entire microbial world was not a good thing, and we wanted to kill and eradicate and stomp out every last bacterium or virus that we could. And it has to do with germ theory. This is one of the earliest pieces of epidemiology. This is a health bill from the year 1664 in the city of London. It runs over the course of a year, and the thinking was, they didn't call it public health back in those days, but that's basically what it was. They monitored and they surveilled and they tracked, why are people dying? What are they dying of? Because the thinking is, if we can figure out what they're dying of, then we can start to prevent those causes of death. And circled in here in red is plague. 7,000 people died of plague this year in the city of London. That's a lot compared to the other numbers on this chart. And what germ theory says is, when we began, we didn't know this in 1664, it would be later in history, around the time of Louis Pasteur, the great French microbiologist, and Robert Koch, the great German microbiologist, that they began to put together single bacterium or virus and a single disease. So there's a bacterium that causes tuberculosis, we know the virus that causes polio, and so on. It's a one-to-one cause and effect. So we thought, oh, geez, the whole microbial world is that way, and we just got to find all these microbes and kill them off. What we failed to realize at that time is that the microbial world is also ecological. It is in relationship with the host organism. It is in relationship with the other microbes in these communities. And so let's fast forward to modern times. And what we have here in the graph on the left is that clinicians and researchers in the post-war years, many of these dread infectious diseases, the ones labeled up here and many more, we began to control those, because once we figured out the single microbe, we could go about eradicating that or bringing those diseases, at least in the Western world, under control. So that's what was done. And how did we do this? Clean drinking water was probably the #1 thing that helped eradicate many pathogens. The other is vaccines are helpful, and antibiotics. My own grandmother almost died from tuberculosis, and she probably would have had streptomycin not been discovered and before it had not been discovered and then made available. So these are all good things. These days, you know, vaccines and antibiotics are still helpful, although there's an awful lot of vaccines that young kids are given. Maybe that ought to be looked at. And you all know how in agriculture there's sometimes inappropriate use of antibiotics, and there's problems with that because of their effects on the microbiome. But let's talk about this slide on the right, the one in color. So you'll notice the time frame is about the same. In the post-war years, we're controlling all these infectious diseases. Longevity, human longevity is increasing. But then it's the chronic diseases that are beginning to take their toll and continue to take their toll on Western civilizations at least. And so these are called chronic diseases because they don't really have an infectious microorganism underlying them. They tend to be autoimmune in nature. They tend to be chronic. Once you get them, you don't really kick them. And it's not just those things on that graph, but it's all of these things too, which are other kinds of chronic diseases. And the hypothesis among microbiome researchers who are looking at this is that what is possibly going on here? And it's this: are we maybe missing some of our microbes? If we're missing microbes and the immune system is not getting the information it needs, then the immune system, which is very powerful, is going to possibly misfire, and it could be attacking healthy tissue. The microbiome is kind of perturbed and scrambled, and it's sending mixed messages to the immune system. And the immune system, if nothing else, it's going to, the presumption is it's a foe unless I get other information, because that's the job of the immune system, is to protect the body that it lives within. There's one thing, I'd mentioned antibiotics. There's one thing, another factor that may be responsible for missing microbes or at least scrambled microbiomes, and that is our diet. Because, folks, down there in the colon, this is not some kind of a garbage can that's on board for our use. That idea is garbage, really. We need to get that out of our minds. The colon is basically your inner ecosystem. It's your inner nature. And when you think about it that way versus, I'm a biologist, I had always thought of nature somewhere out there. It's the woods where we go hiking, or it's the garden. And when I finally began to realize, oh, there's nature inside of me, and many of nature's processes are at work. And that has to do with the fact that this inner nature has living organisms that need to eat. So all of these earlier slides about the life of the soil, there is life inside the colon, and it needs to eat. And in order to understand how diet can affect our microbiome, it's important to just take a quick look at the digestive tract. Here's, down the hatch comes everything. Proteins, fats, and simple carbohydrates are all pretty much digested by the lower reaches of the small intestine. And you can see from that arrow on the left how the numbers of bacteria are increasing as you go down through the digestive tract. It's pretty much our bodily processes and enzymes that can break down that food into compounds and molecules, passes through the gut wall, into the bloodstream, off it goes to where it's needed. But there's one kind of food that we do not, our genome does not encode the enzymes to break down these foods. And it happens to be, goes by several names, you've heard them all, I'm sure. Whole plant foods, fiber, complex carbohydrates. For these purposes, those are all pretty much synonymous. We can't break them down, so they pass into the colon, which is tranquil grazing pastures, really, for our microbiome. They thrive on these poorly digested plant foods. And this is not to say you should only eat plants, because we're an omnivorous species. We evolved to eat all kinds of things. But it is to say, think about your microbiome and whether or not you've fed it on any given day. And so what do we know about carbohydrate consumption, at least in the Western world? On the left there, back in the day, and similar to today, we eat about the same amount of carbohydrates, but back in the day, these carbohydrates were plant foods that were left more or less in their whole form. And so that would be, you all know what a wheat grain is. There's the bran and the germ and all of these layers that make up the bran. These days, what we tend to do with a wheat grain is we strip all of that stuff off. That's the fiber that the microbiome is jonesing for. That is what it lives for. We strip that stuff off, do other things with it, falls out of our diet, and we're left with the sugary part of a wheat grain, the endosperm. That's like the placenta of the wheat grain. And it's full of sugar because that wheat grain is a seed, and that's what gets the seedling going. But it's all sugary for us, and it doesn't feed our microbiome. And so fast forward to today, and basically what we're doing is we're taking whole plant foods and we're turning them into all kinds of other things that tend to be really high in simple sugars, which pass out of the gut around the small intestine, and the microbiota in the colon, they kind of go hungry and malnourished and starving. And that is not a good thing. It scrambles these communities. Maybe we've lost members, maybe they've gone extinct already. And similar to the plant diet, of course, we have a diet, and I'll just draw your attention again to the two biggest arrows on this slide. The problem with the Western diet is there's a lot of simple carbs, and our brain is very, very happy about that. I have to tell you, we've been at these events. I don't know what it is about farming groups in the Northeast. Well, I do know they have some really fine dairy farmers, and they produce some very fine ice cream. And invariably at these events, ice cream is served despite that it's, January to December, and I'm like, my brain, oh yeah, I want some of that ice cream. And so that's what the brain loves and the brain goes for, and there your hand goes for the ice cream. And that's fine. But also ask yourself, did I feed my microbiome today? Before I feed my brain, let's ask, did I feed my microbiome? Because that other arrow in the blue box, the large one on the right, these are all of these beneficial metabolites, which I'll go into, that the human microbiome is churning out in your colon. So it's not, the colon, again, not an onboard garbage can, it is an onboard medicine chest, depending, that is, on what your microbes are consuming. And all of this talk about microbes, what do they look like? What do they look like? Okay, these are bacteria. They've just been colorized to make them easier to see, and they're rushing to the banquet table of fiber in the human gut. This is what's going on. You have a lot of biology and ecology and nature inside of your body. I want to talk about one particular member of the human microbiome, Bacteroides theta iota omicron. Don't say that, you're just going to call it B theta, okay? B theta for short. And those numbers at the bottom, about 300 versus 20. Remember I talked about all those genes that our microbiome brings into our body? B theta alone has genes that encode about 300 different enzymes that do nothing but break down carbohydrates. And there it is, sitting in the colon, doing just that. So B theta is consuming all of these plant foods that you can't digest. And by the way, the cow microbiome, right, which is an herbivore, is a very, very interesting place because the cow is not able to access all of this pasture if you're feeding, if you're doing grass. It's not able to digest all of this stuff. There's a lot of biology in the cow gut, and the cow is largely living on what the microbes that are part of its microbiome are producing. So that's another thing. Again, one of the grandest symbioses that we have, that we know about. So what this tells us, 20, that's the number of enzymes our genes encode. That's not very many compared to 300, and it doesn't allow us to fully get all of the nutrients out of the plant foods that we eat. So B theta, you want B theta. And here's how, let's put this all together. On the left side, you can see fiber, complex carbohydrates coming into the gut, B theta and more, because B theta is part of a community. Eating that stuff up, churning out metabolites. One is called butyrate. Butyrate is chemically, it's called a short-chain fatty acid. And you know what a dendritic cell looks like and what it does. So you're going to put one on the slide right below butyrate on the outside of the colon wall. And that arm of the dendritic cell is going to slide up between the colon cells. It's going to get a sample of that butyrate. It's going to come back down, and it's going to show that T cell there, that green T cell, and it's going to activate that T cell. And it happens that butyrate turns a naive T cell into an anti-inflammatory T cell. And this is a way that diet interacts with our body to control and dampen inappropriate inflammation or inflammation that needs to be brought back down once it's fired up. And had anyone ever told Dave and I this, we probably would have changed our diet a long, long time ago. But they didn't, but now we know, and so mostly what we do now, and I'm kind of the nag on this. Honey, have we fed our microbiome today? I don't know. Okay, well, traveling, it's hard. I'm like, well, this is a great place, this hotel. They've got steel-cut oats. So that's what we're doing. We're eating some complex carbohydrates every day. And it's not just butyrate. There's two other kinds of short-chain fatty acids. This is one of the best studied areas of the human microbiome, these three metabolites here. And the point is, please do not memorize this slide. Just know these short-chain fatty acids on the left, all of those bacteria, including Bacteroides. B theta is there with its other Bacteroides pals in the making acetate and some of these other things. And what do short-chain fatty acids do? That the microbes in our gut make all of these things on this slide. Again, I'm going to summarize it for you. And these are some of the medicinal effects of microbial metabolites in the human body. It keeps pathogens at bay, protects against some forms of cancer, normalizes our sugar and fat metabolism, and it's brain protective. It helps protect against neurodegenerative diseases. And I don't know about you, but to me, this is a big, big part of our physical and mental well-being. And so your microbiome, just like that root microbiome, a plant has personal alchemists too. Plants got them around its roots, probably also on some of the above ground parts of the plant. We've got them around our gut. And it's not just short chain fatty acids, it's all of the compounds listed up here. That is a lot of chemistry going on in our bodies, my friends. And the take home here is that it's estimated that somewhere around 40% of the compounds and molecules circulating in any human body out there at any given time is microbial in origin. In other words, the microbiome churned out that metabolite, not one of your human cells. And so here you begin to see how our inner nature is really sort of something to behold and to think about and to protect and restore and cultivate. And I think it's also worth thinking about all of that in the context of this well-known phrase. Because let's unpack this, right? We've all heard this, you are what you eat. To a certain extent, that's true, but this is more true. We are what our microbiomes eat, whether you're a person, whether it's one of your animals, or your crops, or what have you. All right. I did a little bit of research on Kansas, and I believe it's Kansas Day today. Does anybody know that? All right, so that makes it even better to be here. I like that. And did you know that states, just as there's a state tree and a state flower and a state bird, there is now a move afoot for a state microbe. So Kansans, where are you? Let's get moving on a state microbe, right? So New Jersey has picked Streptomyces griseus. This is a soil bacterium that led to the creation of the antibiotic streptomycin. The hipsters out in Portland, of course, they want to pick Saccharomyces cerevisiae because that's how we get beer and wine. So I want you all to give some thought to what might be a state microbe for you. I don't know what it is in Washington, but yeah, all right. Hey, could we just use micro beer? Possibly. What are some native ferments going on in beer making? And for that matter, I didn't know this until sort of recently, but Kansas used to have wine grapes, right? So what are the native yeasts in Kansas like, and what kind of wines might they produce? You tell me. I'd love to hear. Okay. So here's the new view. Your gut is really a place that is a place of cultivation and thinking about what kind of things you're going to be, what kind of garden you're going to have, and what kind of things are going to be living in that garden and populating it. And knowing that there is a lot within the control of each of us to cultivate our gut. And for Dave and I, we, near writing the end of the book, we're like, oh yeah, this root in the gut, the more we learn about this, the more we see how marvelous these two organs are and how analogous they are when it comes to microbiomes. And part of that has to do with all of the nutrient processing and acquisition. Whether you're stuck down there in the soil waiting for your fetching fungi to bring you something, or whether your microbiome is, waiting for those complex carbohydrates to descend down into the colon and start grazing on them. And microbial metabolites, I've nattered on and on about that, right? This is the other very, very similar thing. We don't even know all of these metabolites produced by the root microbiome or the human microbiome, but it is a lot of chemistry. And then what do these things have to do with? They have to do with immunity and defense for the green body of a plant and also our own body. And I think this adds up to a form of intelligence that we can all tap into and, pull into our daily farming practices, daily lives, because as sure as a root has a rhizosphere, the gut has a rhizosphere too. And I know Steve asked us to talk about the root and the gut, but I want you to think of a brain, and I want you to put a brain on top of the root and the gut as well, because that is really what I'm talking about. Okay, here's your 6 words, your take home. There has been a lot of material that we've given you to think about. Later in the day, you're talking with somebody, or maybe over drink or dinner tonight, and you're like, I can't even remember that. It was just too crazy. All you need to remember are these 6 words: mulch your soil inside and out. Okay, and with that, Dave is going to come up and finish it off. Thank you.
Great. Well, as you've just seen, Anna and I took a fairly deep dive into thinking about the microbiome, both of the plants in our garden and also in our own gut. But I was also left, after seeing how fast that Ann had restored the soil in our yard and literally brought it to life, and having thought about what had happened to the soil in societies around the world, it led to the question of, well, could you do what we had done in our yard and do it on full scale on commercial operating profitable farms in various parts of the world? Now, obviously, as people who live in a city, we were not the right people to actually go ask that question to. So what we did is I took about 6 months off from my job at the University of Washington and traveled around the world to visit farmers who had already restored fertility to their land. And the goal was to basically do sort of, I don't know, maybe call it academic jujitsu, where instead of telling people what to do, I went and listened to them and asked what they had actually done to restore fertility to their land. And what I basically found, and I'll give you the sort of the summary overview, is that these 3 simple principles of conservation agriculture, which are sort of at the heart of a regenerative agriculture movement, really worked to restore fertility to land. Minimal disturbance, or no-till, which is the kind of style of thinking that got me into thinking about this stuff in terms of, well, how do you farm without the plow? Maintaining a permanent ground cover. Basically, that translates into cover crops, both to get carbon back into the soil, but also to keep the ground covered, and adopting more diverse crop rotations. Now, why do these 3 things work so well in combination with one another? That relates to everything you've just heard from Ann. If you were to actually try and figure out a recipe, a simple recipe, for cultivating the beneficial life in the soil, this is what you'd come up with. Give them shelter, don't disturb them. How that first bullet point. What would you do if once a year somebody came, pulled the roof off your house, put in a giant auger and stirred up all your stuff? You'd move pretty fast, right? Well, microbes aren't no different. They need shelter. Maintain that second bullet point. Well, you also need to feed them. Food, shelter, and also community structure. Very few of us would do very well living completely on our own. Again, the example of Matt Damon on Mars does not count. That's Hollywood. One of the advantages of diversifying one's crop rotations, or diversifying the cover crops one uses in one's rotations, are that you get each of those plants is putting out different exudates to feed different communities of organisms in the soil. They're serving different roles. Imagine how dysfunctional a village or city would be if the only people in it were geologists. It would not last. Community structures actually enable us to do more together. And the microbial world is fairly simple. A lot of microbes just have a few chemical tricks. But together, They add up to those millions of genes that Ann was talking about that provide resilience and diversity to the functionality of their host organism. But what I also found in visiting farmers around the world who'd been very successful at applying these principles was that the practices that they used were different in different regions. I visited people from the Dakotas to Pennsylvania to Costa Rica to Equatorial Africa, and the practices people used were very different, not just from region to region, but one farm to the next. But they all adhered to those higher level principles. And I think it's really important to think about the adaptability of those principles to different settings and contexts. There's something like 200,000 different kinds of soils in the world after all. The things that we would do in the Dakotas aren't necessarily going to work in Ghana and vice versa. So those general principles translate, but they've got to be tailored to the specific climate, crops, soil type, economy, technology level, social system that farmers find themselves working on and in. And that's actually really important. We spent a lot of time in the last 100 years trying to take singular technologies and make the land adapt to them. We need to adapt the way of what we do to the land, but using these higher level principles. And if you look at those three principles of minimizing disturbance, cover crops, and a diversity of crops, that's kind of the opposite of how we've taught people to farm for the last 100 years on each point. It's a 180-degree reversal in philosophy. Not a change necessarily in the products that we use, but in the principles we use to think about their application through. This is why I called the book Growing a Revolution. It's a revolution in thought. Now I'm going to spare you the story of all the farmers that I visited today who were so inspirational to me and taught me so much about this. But there were people like Dwayne Beck over there on the left-hand side at Dakota Lakes Research Farm. I visited his farm because if you look at the cover of my dirt book, that's a photograph from the Dust Bowl era from his neighborhood. And I wanted to see what had changed in the last 80 years or so. And he took me on a drive for an afternoon before we went to visit his farm, where we saw only 3 plowed fields in tillage season. They've stopped the black earth from blowing every time the wind blows by going to no-till. They've also experimented with cover crops and diversifying the rotations and have been able to cut their diesel fertilizer and pesticide use by more than half. That resulted in great savings. What happened to their crop yields? They actually went up. They didn't go down. And that was an eye-opener for me, because you often hear when you talk to people in the media that if we go to more environmentally friendly farming, we'll all starve. Well, I've met farmers now who've demonstrated to me that that's a false dichotomy. There's so many assumptions buried in that comparison that need to be unpacked. And Dwayne is a good example of the people who we need to take inspiration from. He was also working with farmers that farmed up to 20,000 acres or so. And I asked him when I left whether the practices that he used would actually work to feed subsistence farmers in Africa, where the real problem of feeding the world for the next 100 years lies, because that's where the next 2 or 3 billion people we expect to arrive on this planet will be located. And he basically said, well, don't ask me that. I'm not a subsistence farmer in Africa. Go talk to that gentleman on the right, Kofi Boa, who runs the Notill Research Center in Kumasi, Ghana. He's another inspirational figure who's in learning to apply the principles of conservation agriculture to his neighborhood, have taken the farmers he works with from their traditional slash and burn style of agriculture to using no-till with cover crops. They get their diversity in all at once, because this room would actually be several farms in the community that he works with. And so they will grow 6 to 8 different crops in the field at the same time, because it's almost all manually worked. They have a totally different technological application, but it worked for them in terms of doubling and tripling their corn and cowpea yields inversely. I tripled corn, doubled the cowpea, if I recall correctly. That's as good as the Green Revolution did. But why did the Green Revolution not work very well for many subsistence farmers around the world? Because they don't have money to buy fertilizer. They don't have money to buy patented seeds. They don't have money to buy pesticides. What they have is a little bit of land and their labor and a lot of ingenuity. Kofi worked his people through thinking about the soil differently as a way to greatly increase their output, which transformed his village economically. You all probably also know Gabe Brown. In fact, I think it was at No-Till in the Plains that I first met Gabe Brown. So thank you all very much for that introduction. He's a very inspirational figure, and he has a new book out that is fabulous. I recommend it. I know maybe I shouldn't do that as an author, but but it's a great book. His story is truly inspirational, and there's a chapter on him in our book too. But But he was doing stuff in terms of bringing livestock back into his cropping operation and also using them as a tool of soil restoration on the native prairie he's trying to bring back. His example really changed my thinking from thinking of cows as an agent of soil erosion and land destruction, for which you can find plenty of examples in the world and that I've worked on and published on before. But he taught me that the problem is not the cows. The problem is how we manage the cows. The problem was how we think about how we use livestock. David Brandt, the gentleman on the right, is another inspirational figure that I met. Grows corn, wheat, and soybeans for the commodity markets, but he also grows diverse cover crop mixtures. First field I think I sort of stepped in that had like 12 or more different cover crops on it at the same time was the one in that photograph where he's holding up and modeling his tillage radish. Notice the yellower field behind David's field. That's his neighbor's soybeans. Look at the little patches of green in there. They may occupy up to maybe about a quarter of his neighbor's fields. Those are glyphosate-resistant mare's tail weeds. They've been sprayed 3 times that year. You can't kill them. They're a yield drag on his neighbors. Part of what started me on the road to optimism about being able to restore fertility to the world's farmlands were stories like David's, where he's now much more profitable than his neighbors. In fact, he just bought a big section of his neighbor's farm. And how is it that David was able to be more profitable than the people right next door? He had a totally different management practice. He's been no-till for 44 years. He's been doing cover crops for a while now. He uses so little nitrogen and glyphosate that I teased him that he's an organic-ish farmer, which he thought was really funny. I thought it was really funny. It was great. But it's deadly serious. By using fewer inputs and maintaining a higher profitability, he was able to restore fertility to his land. The soil over there on the left, this is a slide that he was kind enough to provide me with. He's farming a heavy clay soil, the Cardington clay soil. The slide on the left labeled 2014 is the soil that he had about the year that I visited. I visited in 2015, so that's about what he turned it into. The one on the right was, I believe, from his neighbor's farm that is very comparable to what he started with back in the early 1970s. This is like that tobacco plantation comparison I showed you. This is like what Ann did to our yard. But David did it with intensive agriculture, profitable intensive agriculture. This is the cause for hope. And each of these people that I've shown you has done something like this. If we can turn what's known today as conventional agriculture into something that, instead of degrading the fertility of the land, builds the fertility of the land over time, that is a true revolution. That could be a true revolution. And what Gabe Brown was doing was bringing his livestock back into the picture, was using them as essentially self-propelled methane digesters to harvest his cover crops and crop stubble, turn it into manure, and return that to the land. What did that actually do? Well, the slide on the right is the soil from that field that I just showed you. It's sort of the best of the best off of Gabe's farm, where he has his market garden and cover crops, and then the livestock comes in. Nice, rich, black earth. Looks a lot like probably what the native soil was like in this area. Notice in his left hand, that's his neighbor's organic farm. It's nowhere near as nice. What's the problem with the organic farm? Well, it's not that they use too much nitrogen or chemicals, it's that they plow too much. This set of principles, the no-till, the cover crops, and the diversity of rotations, they go together. I've parsed some of the studies that are out there in the literature on carbon sequestration in the ground and building up carbon in the soil organic matter. It's the combination of the three that result in rapid change. If you just go no-till without the others, it takes longer. What I basically was able to put together is a phrase that Anna helped me a lot with in trying to strategize of how to describe the Growing a Revolution book, the recommendations from it. And what we came up with is ditch the plow, cover up, and grow diversity is the simple recipe for enhancing soil health and the fertility of the land. Now, I would put a little asterisk on there now and saying yes, and maybe accelerate it with livestock, because I view livestock as an accelerant, a potential accelerant on this process. It's not necessary, I don't think. I think people who tell you that livestock are essential for building soil health, well, I think that's not actually right. It could just take a lot longer if you don't have it. If you want to do it fast, I'd bring the livestock in, but adhering to those three principles. And I also want to emphasize this is not stuff that Ann and I are just making up. There's studies coming out in the peer-reviewed literature that back up the style of science that you were just seeing that she said. So I'm going to run you through a few examples of those. My favorite one is the paper by Claire Lacan and Jonathan Lundgren that came out, I think it was last year, where they went around to conventional and regenerative farms around the Midwest, and they looked at profitability. They looked at the total cost, sort of the revenue per hectare and cost per hectare. So the total bar is revenue. You then subtract the cost for all the different things in the colors that are enumerated on the right. But notice the white bar left over at the bottom. That's your profit. Regenerative practices were more profitable across the board. It's not just that David Brandt's figured out how to do it or that Gabe Brown has figured out how to do it. It's that these practices actually work. When you spend less to grow as much or not more, you have a better bottom line. And even an academic geologist can recognize the simple economic truth of that. The thing that surprised me more, though, was that Jonathan is an entomologist. He studies insects, the way that Ann has plant lust. Jonathan apparently has insect lust or something. Oh, that's probably, you shouldn't say that out loud, huh? But notice that basically he's looking here at corn pest abundance on regenerative farms versus conventional farms. The conventional farms are the ones using the pesticides. What's the difference? They have 10 times as many pests. Do you think if they put that on the label that anybody would buy any? Increases your pest abundance tenfold. No. So what is it about that? Well, because basically broad spectrum pesticides kill off the things that eat your pests. And so if you have other means, other ecological means, in the scientific, not the political term, if you have other ecological means of managing your pests, you can actually spend less and do better. There was a paper that came out of Japan called The Critical Decline of Earthworms from Organic Origins Under Intensive Humic Soil Organic Matter Depleting Agriculture. This gentleman who had a fascination with earthworms apparently put together a global survey of what's happened to earthworm populations under conventional agriculture. And the answer is they've been decimated. I wrote an article for the Daily Beast that they titled The Earthworm Apocalypse. You don't get to write, and it's a simple note, if you write for publications other than your own books, you don't get to pick your headline. You get to live with it. There's a paper out of the Journal of Plant Nutrition back a few years ago, a decade ago actually, where people were looking at to what extent that if you have arbuscular mycorrhizal fungi associated in this case with an onion crop, what does that do to phosphorus uptake by the plants out of the soil? Notice the numbers where you go from 0% mycorrhizal colonization to 100%, you increase the phosphorus uptake tenfold. By some estimates, there's enough phosphorus that's insoluble that doesn't show up on conventional soil tests in the world's farmlands to support farming for another 50 or 100 years without adding any more anywhere. How would you do it? Put the fungi to work. This is what Ann was talking about. How do you kill all the fungi? You plow them and you apply a lot of nitrogen. Another paper, this one came out of Berkeley, where Ann and I met at grad school. And it came out last year. The title formally was Nutrient and Dose-Dependent Microbiome-Mediated Protection Against a Plant Pathogen. That's the kind of thing you have to title stuff in journals. When it got out into the popular media, what it was translated into was fertilizer destroys a plant's microbiome's ability to protect against disease. This again is what Ann was talking about. I don't think it's any coincidence that global sales of pesticide went through the roof after global sales of fertilizers. Because we dismantled the natural defensive systems of our plants. And that's not to say that all fertilizers are bad. If you want to build soil organic matter fast, if all you do is grow carbon-rich sources, you got to find some nitrogen to combine with that to turn it into organic matter in the soil. And there may be cases where adding nitrogen fertilizer is the best way to do that. The point is that we don't need to add as much of it as we've been adding, and let alone the fact that only half of it actually gets into plants. There's also been studies that have looked at how the soil microbiome is more heterogeneous, more variable and organic than conventional farming systems. I would love to see studies that actually compared regenerative and conventional systems side by side in this regard. I think we would see very similar results, but I can't find the studies. There's very few that are actually out there in the literature. But the point is, there is science being done that backs up the kind of stuff we're telling you about. You'll notice none of it was USDA, none of it was land-grant. Go back and look at the places I was just talking about. I think that's a shame. What are some of the benefits of rebuilding healthy, fertile soil? higher farmer profits. Any form of sustainable farming has got to be profitable for the farmers or it won't be sustained, and full stop. But it also can produce comparable yields. This idea that that we will starve if we don't maintain our current practices is fiction. We can use less fossil fuel, less fertilizer, less pesticide to grow as much. We can put more carbon in the soil. The differences between all those khaki soils and the dark soils we showed you, that was organic matter and carbon. It helps with water retention, helps limit off-site pollution, which can help clean up water supplies and so forth. But there's one other issue that I'll use the last 2 minutes that I have to basically illustrate to you. And that's basically the connection to our own health. Because the soil is where our food comes from. And there's ideas that go back to the 1940s when Lady Eve Balfour suggested that poor soil health translated into sick crops and farm animals and sick people. She didn't have much in the way of mechanisms she could identify in terms of the soil health and civilizations. I've written about that. But there have been documented declines in the mineral abundance in foods over the last century or so. There's various studies in various amounts, but you just notice that those numbers are anywhere from 16 to 76%. You know, they're not small numbers. We're not talking about a few percent changes. And they would be different for different crops in different areas and so forth. And notice that for beef, it's got about half the iron it used to have in it. And what about the phytochemicals, the secondary plant metabolites, which are some of the things that Ann was talking about, plants making that help with their defense. Things like linalool in lavender, sulforaphane in the brassicas that has anti-cancer properties. Things like curcumin that's in turmeric. These things, how much have they changed as a result of agronomic practices? Well, if you look at the meta-studies that are out there that compare organic versus conventional produce, and that's about the only dimension there has been. I want to see studies comparing regenerative versus conventional as an additional dimension. What they always find are differences in vitamins, antioxidants, and phytochemicals. Results are pretty mixed in terms of the macronutrients and the mineral micronutrients. And I think that's because if you look at the soil health on organic farms, it ranges from terrible to great, and you look at the soil health on conventional farms, it ranges from terrible to great. That's why I have apples, oranges, and bananas on the screen. A lot of these meta-studies aren't, I think, comparing what needs to be compared, which is food grown in healthy fertile soil and food grown in degraded soil, in both, whether in organic or conventional systems. And the phytochemical and nutrient properties and the contents of vitamins, phenolics, and antioxidants are generally higher in organically produced fruits. I'd like to know whether this is also true in regeneratively produced crops. In other words, to what degree can we connect healthy soil to healthy crops to healthy people? That's essentially the book that Ann and I are starting to try to work on now. We're looking for data that people may have. And if anyone here has data on these kind of comparisons, please feel free to share it with us. We're trying to find as much as we can. I'll show you two very quick examples before I end. One is from a master's thesis in the Netherlands, where they basically compared mineral density of crops grown in conventional farms and organic farms that use non-inversion tillage and cover crops, close to the conservation agriculture idea. They found large differences in mineral densities between those systems in a very short time. The one example that I'll show you and end with is an example that Ann and I did in our sort of experience in the lab, where there was a farmer in northern Oregon who had done a 2-year side-by-side cover crop comparison. The land had been no-tilled for a while, so he's adding the cover crops. And one was just a chem fallow rotation, and the other had a diverse cover crop mixture for 2 years. We sampled his wheat in the second year, same cultivar, same soil, side-by-side fields. What this shows you is what we got after grinding up his wheat, dissolving it in acid, and running it through the mass spec. All the mineral micronutrients over there are on the left. The cover cropped amounts in parts per million are shown in the first column. The conventional ones are the second column. Ignore that and look at the third column. It's the ratio of the cover crop to conventional for each element. I've highlighted in green zinc. Why zinc? It's really important for fighting colds, and that's something that's on the top of my head right now. Notice the difference, 56% more zinc. You can't change the soil that fast. There's just as much zinc in those two fields. What's different? You can change the biology that fast. You can regrow the mycorrhizal fungi. You can get those microbial miners Ann was talking about back to work in the soil to get that out for you. If the way that we farm, if regenerative farming can produce healthier, more nutrient-dense food, people in cities are going to pay a lot of attention to that. They're going to want it, they're going to demand it. We need to study more about this and understand it better, because the science is getting to the point where we may be able to. And I'm going to stop there. I think I'm the guy standing between you and lunch, if I know the time correctly, and I don't like being in that position. And I did bring books with us. We're all the way down at the end of the hall if you want to come by and check it out sometime today. But anyway, thank you for what you do, and we'll get on to lunch.
Yeah, don't wait.
Well,